Good day all. I am Fadipe and I'll be presenting on behalf of my co-authors the poster titled Growth Patterns of Novel Polyhydroxyalkanoate Bacteria Isolated from Oil-Contaminated Site Soils in Some Selected Carbon Sources. Here's our poster. Polyhydroxyalkanoates, for short PHAs, are biodegradable polymers accumulated intracellularly by a wide variety of bacteria under environmental stress conditions, which leads to unfavorable growth patterns. They are made as energy molecules so that when conditions of growth revert to normal, they can be converted back to acetyl-CoA and directed towards energy generation. They can be completely degraded by organisms containing PHA depolymerases to carbon dioxide and water. Thus, they are eco-friendly. They are classified based on the number of carbon atoms in their functional groups into short chain length, medium chain length, and long chain length PHAs. Their chemical and physical properties are quite synonymous to those of synthetic polymers such as polypropylene and polyethylene, and as such, they are considered suitable alternatives for bioplastic production. A wide number of bacterial species actually manufacture PHAs under condition of limiting nutrients and high carbon source in the medium. And the most important enzyme in the biosynthetic pathway is the PHA synthase. PHAs are found application in medicine, biomedicine, in food, in packaging, and in the production of paints. In this work, we were interested in isolating polyhydroxyalkanoate producing bacteria with capacity to utilize varied carbon sources. As a result, we isolated bacteria from soils of oil-contaminated sites. We collected samples from Dalico market sites coded D and Mechanic Village site coded M. We isolated bacteria on nutrient agar according to the serial dilution and populate methods, and we obtained pure colonies by strickling. Colonially distinct isolates were selected for further studies, and their colonial characteristics, cellular morphologies were observed. We also observed their reactions for catalysis and starch hydrolysis. These isolates were screened for PHA production on nutrient-limiting carbon-rich medium using glucose, glycerol, starch, and olive oil as carbon sources, with the viable colony method using now red and now blue hay, and further by Sudan black bee staining. These isolates were used for PHA's production. Actually, 4 out of the isolates were selected, and their growth patterns were observed during PHA's production. 3 of the isolates were selected for further work and were characterized by 16 sRNA sequencing. Our data was analyzed using sequential for sequence assembly, sequence homology and alignment was done using NCBI BLAST-M tool and Cluster W respectively. Phylogenetic analysis was done using the Tamura 9 method of the neighbor joining algorithm in Mega 7 software, and other data were analyzed using MS Excel and SPSS version 20. With respect to our results, we had a total of 27 colonially distinct isolates, 11 from the Mechanic Village site and 16 from the Dalico market site. Out of these isolates, a total of 25 were gram positive and we had 2 gram negatives. 25 were rods and 2 were cocoid rods. All isolates were positive for catalase tests, 17 were positive for starch hydrolysis, and 10 were negative for starch hydrolysis. The screening results is displayed in figure 1 to figure 4, 1, 2, 3, and 4. And we can observe fluorescence under UV light in all the plates for the isolates. 6 isolates were selected from this screening for further studies based on their capacity to utilize all the 4 carbon sources and they are isolate D23A coded 19, isolate M11B coded 26, isolate M11C coded 27, isolate M14A coded 1, isolate M21A coded 14, and isolate M21B coded 15. And you will see that they produced fluorescence, orange fluorescence, blue fluorescence, and we had even some that had some yellow fluorescence in addition to the orange and blue fluorescence in the plates with now red and now blue here. We also observed in figure 5 the summary of the screening result that glucose, glycerol, and starch, we had a lot of organisms that were positive on those carbon sources. We had about 40% organisms that were positive with olive oil. And we observed that glucose was the carbon source where we had most pH positive isolate. This is not surprising because we know that most organisms, living systems, prefer simple sugars such as glucose for energy generation. Out of the 6 isolates, 4 were studied for their growth patterns, and the results are shown in figure 6 to figure 9 here. So figure 6 is for M21A, figure 7 is for D23A, figure 8 is for M11B, and figure 9 is for M11C. So we observed that M21A, D23A, and M11B utilized glucose best as a carbon source, followed by glycerol, olive oil, and starch. And for isolate M11C here, it utilized olive oil, followed by glucose, followed by glycerol and starch. In summary, we found that isolate M11B was the best utilizer of glucose, glycerol, and starch for growth and pH's production, and isolate M11C was the best utilizer of olive oil for pH's production. 3 of these isolates, D23A, isolates M11B, and isolate M11C, were characterized by 16S RNA sequencing. The figures of their phylogenetic analysis tree are shown in figure 10 and 11. So we found that D23A, which is now coded P1, clusters with Lystinobacillus species, and they are indeed Lystinobacillus species, a strain of Lystinobacillus species. We found that isolate M11C and M11B, coded as P3 and P2 respectively, are strains of Pseudomonas aeruginosa, and they clustered perfectly with other strains of Pseudomonas aeruginosa species. In summary, we've been able to isolate 3 pHs producers with capacity to produce pHs from this work. To summarize our findings, we isolated 27 bacteria, 23, 21, 19, and 12 were positive for pHs production on glucose, starch, glycerol, and olive oil. They produced orange, blue, and yellow fluorescence, and we had blue-black intercellular bodies observed. And we found that M11B was the best utilizer of glucose, glycerol, and starch, and M11C utilized olive oil best. 3 isolates were finally selected, 1 Lystinobacillus species and 2 Pseudomonas strains. With respect to further work, we intend to use these 3 strains for optimal pH production using carbon-rich agro-industrial residues and to improve them for optimal performance. These are some of our references. And to conclude, I would like to appreciate the members of the PHS Biotech Task Force, my undergrad interns and my graduate interns. And these are the members of the PHS Task Force for the amazing job that we've done on this project. We would be more than happy to have further discussions on this work and to answer questions. And this is my email address displayed here. We welcome this for further collaborations. And I would say thank you very much for listening.